The main question being addressed in Figure 6 is what are the effects of short-chain fatty acids on blood-brain barrier permeability? In the first experimental design, Part A, it investigates the effects of specific short-chain fatty acids on the blood-brain barrier throughout different parts of the brain. They injected the germ-free mouse with monocolonized bacteria to see if it penetrates the permeability of the blood-brain barrier. They picked sodium butyrate to continue this experiment because sodium butyrate produces more occludin, which is one of the main blood-brain barrier's proteins.
For part B through D of the experimental design, they picked sodium butyrate to continue this experiment because butyrate is one of the main short-chain fatty acids, also because sodium butyrate produced high occludin and it mimicked the BBB, blood-brain barrier, of pathogen-free. They then used a Western blot test to compare the germ-free and sodium butyrate in different parts of the brain to detect the specific proteins that were extracted from the sample.
For the methods being used, there was two main parts of the experiment. The first was the Evans blue dye, and then the Western blot and the protein extraction. For the first part, we have the Evans blue dye. To set up the experiment, they took 5 mice, 2 of which were your controls, the pathogen-free and the germ-free. In order for the blood-brain barrier to work, certain proteins need to be made. The pathogen-free should have a fully in contact blood-brain barrier, and the germ-free shouldn't produce the bacteria that should make that. C-but and B-theta are both monocolonized bacteria that they injected into the mice, and both of those bacteria produce parts of the blood-brain barrier, certain proteins for the blood-brain barrier. For Nabu, that was given a bacterial metabolite of sodium butyrate, and this is the specific protein for the blood-brain barrier that it needs. They anesthetized the mice and they put the things in there. In the figure, you will see the results for the Evans blue dye. The red represents the damaged BBB, and so in the germ-free, you can see that it's not very. It didn't stop a lot, and it leaks a lot more. And then the blue represents the nucleuses. So if you see more blue, that means it should be more in contact. For part 2 of the experiment, they took the sodium butyrate and the germ-free, and they did a Western blot that compared the production of certain proteins, specifically occludin and claudin 5. B-tubulin is your control of this, and the occludin and claudin 5 are protein parts of the blood-brain barrier that are important. And so here you just compare 3 different samples, one taken from the frontal cortex, the hippocampus, and the striatum. And so after extracting the protein, they ran it through the Western blot. And then after they did the Western blot, they ran it through a program that told them a quantified number of how much they have.